This is the introduction to lecture 4. We just finished talking about the structure of DNA and how DNA is replicated. And we are now going to talk about how genes are transcribed to make messenger RNAs or RNAs that are complementary to them and are part of the whole procedure for how the information in a gene, which is DNA information, gets converted into the information to produce protein, which is an amino acid sequence. So that's the focus for this part, is the actual transcription part. And these are the learning outcomes for this section. So I would like you to be able to describe the differences between RNA and DNA. They're actually remarkably similar. There are some very key important differences. And to be able to distinguish the terms when we're talking about, and this actually relates also to when we start talking about translation, because it's key to translation as well. But just different ways of describing the DNA strands of a gene. There is the template or antisense strand, and the other strand is the coding or the sense strand. And knowing what these refer to, you will find them in different courses. You'll find them discussed a little differently. When we talk about transcription, we talk usually about the template and the coding strands. Yes, but the sense and the antisense we tend to talk about more when we're talking about sort of working with DNA and with genes and that sort of thing. So you will hear them used differently, and yet they do mean the same thing. So this and then these are the same. And then any term that I've bolded needs to be explained in your own words. So if I had put template strand, in bold, which I should have done, then you would be able to explain that in your own words. If it was an enzyme like RNA polymerase or something like that, some kind of factor like the sigma factor, that includes what the function is. What does this do, and sort of when does it act, and that kind of thing. And be able to identify sort of core promoter elements, which are regulatory DNA, what the transcription unit is, and how that differs from the coding sequence, and so on. There's an image of a kind of a full gene with all the sequences and then different subparts of that gene, and I'd like you to know those. And then the processing. What I should also have included, and I will, is just to be able to outline the steps, right? So processes of DNA replication, of transcription and translation all have an initiation stage. So how does the process get started? And then elongation. So how do you add more nucleotides or add more amino acids? And then how do you terminate the process? So we want the steps of transcription in the at the level basically covered here. I'm just going to put a little in there. Okay, and so this is this conceptual framework, which I kind of left you with last time, that DNA provides the information to produce RNA, and that carries that information to the place where translation occurs to make protein, remembering that there are also some functional RNAs, tRNAs, ribosomal RNAs. These are part of the translation process, and the RNAs do a lot of the actual catalytical work of those processes. And the DNA replication, of course, leads to meiosis and mitosis, which we are going to talk about later. So this is the whole unit of the molecular side of things, sort of encapsulated in one simple diagram. So we've done this part, and then this is kind of where we are here. So the instructions from the actual DNA has an intermediary, which is an RNA molecule, and transcription is the name of the process that produces that. and they provide that information to for the ribosomes and the tRNAs to read and then produce amino acids. And in prokaryotes and eukaryotes, they are somewhat different. In the prokaryotes, such as bacteria and so on, they don't have a nucleus. So the process of transcription and then later translation, they're not compartmentalized. So what's really interesting is while the RNA is being produced by transcription, that's what this red line is here, um, the the ribosomes can just jump on and start translating before the transcript is even finished. So you can get these really rapid responses. Bacteria can respond very quickly to environmental changes, and that is because the genes that they transcribe in response can be expressed very, very quickly. In eukaryotes, there's more to it because the DNA is housed in the nucleus, which is a separate little compartment in the cell. The transcription takes place there, the messenger RNA copy is made, and then there's a series of processing steps that we'll talk about a little later in the lecture that have to occur before the message is complete. So the mature messenger RNA is then transported specifically out of the nucleus into the cytoplasm, and it's out in the cytoplasm where the ribosomes, bind and undergo the translation process. And so these processes are separated into different compartments, and they, eukaryotic cells can still respond to things, fairly quickly, but there is a little bit of a delay because of the extra steps and things that have to take place. RNA and DNA have very few differences. One of them is that the sugars are different. They are both ribose sugars, and this is deoxyribose, and hopefully you remember the carbon. So that's the first, second, third, fourth, and fifth. Just to note, when you see diagrams like this, the corners, they often don't put the carbons in there. And when we look at uracil, you'll see the same thing. And so this is DNA, and remember the deoxyribose is this carbon 2, the 2' carbon, because it has a hydrogen rather than an oxygen. Ribose has a hydroxyl group there. And so this is the one difference in the ribose sugar between DNA and RNA. And it does make the RNA a little bit more reactive than DNA. DNA is a little more stable. But the other main difference and the other main reason that RNA can be less stable is it's single-stranded. That doesn't mean it can't fold over and base pair with itself, because some RNAs do that. You'll see that when we talk about tRNAs in a couple of lectures' time. But being single-stranded means that it's a little bit less stable as well. Okay, and then the other difference, as I said, you'll see that chemically they're very, very similar because they have guanine, cytosine, and adenine, just like DNA does. But instead of thymine, and this is what thymine looks like, and again, the little corners on these diagrams are carbon atoms. They're just not included. They're, people who are used to looking at these know that those are carbon. So that's just part of the secret language of sort of nucleotide chemistry and other molecules too. But in thymine, there is a CH3, a methyl group on this carbon, whereas with uracil, this is one that shows the carbons, and then this is one that's similar to this diagram. And unfortunately, it's reversed, but it's right here that this is. So there's no CH3 group right here on the uracil, and that's the other chemical difference. But aside from that, let me just erase that so it won't be confusing. Aside from that, uracil can bind complementarily with adenine with 2 hydrogen bonds, just the same as thymine does. So you may remember from the previous class we talked about A and T having 2 hydrogen bonds between, and G and C having 3. And that means that the GC pair is slightly stronger binding than the AT pair. And it turns out that the A and the U pair is the same. It's got just the 2 hydrogen bonds. So it's a slightly weaker bond than the GC pair. And we'll probably talk about that binding a little more later, or even in some of your other classes when you start talking about PCR and some of these other methods, the number of hydrogen bonds between the complementary pairs is significant in figuring out what conditions to use for those techniques and so on. Now, not all genes are translated, are encoding proteins. Some of them encode RNAs that just do their job as RNAs, and there's a few that are of significance for us. So tRNAs, as you know probably already, are responsible for bringing the right amino acid to the mRNA while proteins are being synthesized. So they're kind of like carrying the amino acids and onto the ribosome, and then in the ribosome, RNAs, ribosomal RNAs, are major components of those ribosomes, and you would think maybe that it is the protein part that does the actual connecting of the amino acids together, but it is the RNA component that does that. And there's also small nuclear RNAs. These are parts of what are called SNRPs, small nuclear riboprotein particles. So these are complexes of proteins and RNAs, and they are involved in the splicing of RNA introns. And we're going to be talking about splicing at the end of the prep, but I will be dividing it into some smaller segments so that you can, go through it in smaller doses. And there are others that you'll probably learn about in some other courses, that some of the small RNAs that are involved in regulating genes and mounting responses against viral infections and that sort of thing. They do a lot of interesting things, and those are for other courses. Okay, so RNA is produced through the transcription process, and in transcription, the DNA of a particular gene open up. They separate, they unwind so that the DNA has a little bubble. It's very much like, it looks like the replication bubble that we talked about last time. But in this case, just one of those strands is going to be serving as the template, the DNA that gets copied for RNA synthesis. And incoming ribonucleotides bind to the DNA nucleotides on the template strand. So if you've got an A on the template strand, there's going to be a U on the RNA strand that's being made. And if the next one is a C in the DNA, then a G is added, and so on. So they're added that way. And there are, of course, always going to be a few exceptions, but for the most part, unlike the DNA replication process, when you have your replication bubble, this is a very bad diagram, but that's the idea. And you're producing new DNA. You're producing it on both of the strands, leading and lagging. Whereas in the case of transcription, again, there's going to be, you may even learn of some exceptions in other courses, but for the most part, it's just one strand that's being copied to produce the RNA. And that strand is going to be the template strand. Yes, and the messenger RNA is usually called a transcript, so that's sometimes it's called the message, sometimes it's called the transcript. When we talk about these things, we tend to have a number of different names for them. Also messenger RNA, mRNA. And so on. Okay, so here's a schematic. This is your DNA strand here. And so notice it's opened up and there's what looks a little bit like a replication bubble. It's called the transcription bubble because that's sort of what it appears like. This is the RNA polymerase. Now the RNA polymerase is not just one protein. It's a complex of 6 proteins, and some different factors do different jobs. We're really only going to look at the holoenzyme, which is the component that has 5 subunits, and then the sigma factor when we talk about prokaryotic transcription. But this is the idea. You've got your RNA polymerase, and it's moving. Let's see. This is the RNA that's being produced. So it's produced from the 5' end to the 3' end. So the RNA polymerase is moving this way along the DNA. And as the RNA polymerase moves along, the template strand opens, so it's unwinding here. The enzyme is moving along very fast. I'll tell you how fast it is in shortly. And as the RNA is produced, nucleotides that are complementary to the DNA are made. Okay, so this is proceeding in this direction, 5' to 3', just like it does with DNA. And then as this is opening up and the enzyme is moving in this direction, the DNA then winds back up behind the RNA, and the RNA part gets sort of pushed aside, displaced, they say. So these two guys, these two pieces of the DNA bind to each other again, wind back up, and then that pushes the RNA kind of out. Yeah, so sometimes you will see these. I'll probably post some animations and little short videos that just show the process animated, because it's sort of interesting to look at that way. And you'll see it looks like a ribbon is just coming out of the DNA, and the ribbon is the actual RNA part. Okay, so as I said, RNA is synthesized in the 5' to 3' direction, exactly like DNA. So as the transcription is going along, new nucleotides keep getting added on the 3' end, and that's what we mean when we say 5' to 3' direction. Notice that as this is happening, it's reading the template strand at the 3' end to 5'. Okay, so it reads at 3' to 5', but it produces RNA 5' to 3'. Now, RNA polymerase, unlike DNA polymerases, no primer is needed. The RNA can be made de novo. De novo means newly or by itself. It means you don't need something to add nucleotides onto pre-existing. The RNA can just, the enzyme can be here, and it can just start making RNA that's complementary to the DNA strand on the template side. So each gene is for the most part transcribed in only one direction. But on any particular chromosome, each different gene has its own direction. So some of them will be transcribed. So if you suppose you were to unwind like a section of a chromosome and it had 3 genes on it, there's no reason to think that all 3 genes would be transcribed in what we call the same direction. So the template strand wouldn't necessarily be the top strand for all of these. In some cases, the top strand, this one, is the template strand, and in other cases, that's the template strand. And so this is just meant to show that for different genes, so for each gene, the transcription direction is always the same, but for this gene, it might always be this direction, and for this gene, it might always be from the other strand, the other direction. So each one is kind of regulated separately, and it could be either a strand that is the coding strand along a chromosome. A lot of students feel like if you looked at a chromosome that like the coding strand is just one of the whole strands of the whole chromosome, but it's different for each gene. So this would be template and coding, this would be template and coding, and this would be template and coding. So these terms are specific to each gene, not to an entire chromosome. I guess that's what I'm just trying to be really clear about. Okay. So overall, at the initiation process, the DNA has to be unwound. And then this is a better example of, so there's two genes here, and this gene is being transcribed in this direction because the RNA is being made starting here, and then the new nucleotides are being added on the 3' end. And then this one is being transcribed in the other direction. And notice they're right next to each other on the chromosome. So each of them has their own direction of transcription. And also, this also just kind of points out what I was saying about how the DNA unwinds ahead of the RNA polymerase, and then it rewinds and closes up the double helix again. So the transcription bubble doesn't get any bigger. It looks exactly the same size as the polymerase moves along the template. And same over here. We're rewinding here and we're opening up here because the polymerase in this case is going in this direction. So for some of these things, just trying to think through the process, and then you may have to go through it mentally a few times to feel that you're comfortable with it. If this is new information for you. Some of you have probably covered this kind of material before already. And then the complementarity when the RNA is being made. So far we've just been showing, this is the first one that really shows sort of the actual nucleotides, but just notice the complementarity. So what you have on the DNA, the complementary nucleotide is put on the RNA. And that's shown here as well. So each new one that comes is added onto the 3' end, except for the very first one that was just placed there. It will be complementary to the DNA nucleotide, and this is the template strand. Okay. So the template strand of the DNA is the one that's copied to make an RNA that is the reverse complement. Reverse complement means it has the complementary nucleotides, as you can see here, and the polarity is opposite. So remember that when the nucleotides bind to each other, they can only do so if they're aligned backwards relative to each other. So the 5' and 3' have to be in opposite directions. And by convention, we usually, when we draw DNA and we're trying to indicate polarity, the arrowhead usually means the 3' end, and the flat part of the arrow usually means the 5' end. That's just a conventional way that we do it. So if DNA is given this, an arrowhead, then you know that's the 3' end. And so what that means is the template, so the template DNA is like this, and then the messenger RNA is like that. And that's what we mean when we're talking about reverse complement. The reverse refers to the polarity. Now notice the other DNA strand. It has practically the same sequence as the messenger RNA. Okay, and that's why it's referred to as the coding strand. The messenger RNA has the nucleotide information that's going to give rise to the protein structure. So it has the instructions for the protein, and so it has what we call the coding information. It's also referred to as the sense strand because sense refers to the fact that it has, again, the information for the amino acid sequence. So coding and sense means the same thing. Non-coding, or sorry, coding and sense is this. Non-coding is this. This doesn't have the it does have the amino acid if you reverse complement it, but basically this is the DNA that was the template strand. And it's also sometimes referred to as the antisense strand. Now, we tend to use this term, antisense, when we're talking about RNA for different kinds of molecular uh, applications, procedures and stuff, but you may sometimes hear even the DNA referred to as the antisense strand. The antisense because it's the reverse complement of the sense strand. Okay, so hopefully that makes sense. And then you want to just go through and make sure that these questions can that you can answer them, because if you can answer these things and you can say what the RNA, how the sequence compares to the template strand versus how it compares to the coding strand, and then why then we call the non-template strand the sense strand and the other one the antisense. So just if you understand why these are named the way they are, then you will have understood this part. This is the part that's most important. If you were to go on to do any kind of molecular work, this kind of understanding of DNA and RNA and coding and non-coding and working with genes and stuff is really important. Okay, and so these are the terms, and I did remember, after all, to bold them. So those are the ones you need to know. And so transcription, just like replication and, as we'll see, translation, has three phases. One is the initiation phase. So where does the transcription start? You have to have some kind of way of knowing where a gene begins and where it ends, and what part needs to be transcribed and what part doesn't. And then elongation is just the repeated step. So each time we add an extra nucleotide, we have made the RNA longer. So that's the sort of iterative, repeated steps of adding nucleotides. And then termination. At some point you have to stop because the gene is finished, and you don't want to go beyond that. So how is that done? And we will come and begin again in the next prep at the initiation, and talk about elongation and so on.